Okay, class. So this is lecture 8, and I'm going to call this lesson 10 because it's the 10th video that I've made. But this is going to cover the lecture 8 PowerPoints. And a lot of this is just going to be review. So we're just going to go back over what we learned from Mendel, right? So what did we learn from Mendel's pea plant experiments, right? So Mendel said that they're heritable units, right? He called them, well, he called them heritable units, but we call them genes. And those controlled a single trait, right? So we talked about is the seed round or wrinkled? Is it yellow or green? Is the flower purple or white, right? And so that's what we're talking about. We have this gene that's controlling a specific trait. So in this case, one of the examples we had was flower color. And so we'll call that the gene. And we know that there's alternative forms of the gene. And we know that in this case, we have purple or we have white. And we did the Punnett squares, and we know that this particular allele is purple. That means its DNA codes for an RNA that makes a protein that folds into a certain shape that reflects light that's purple in the purple wavelength. And we know this one is white, right? Because it's broken for whatever reason. It's not folding into that particular shape. Maybe it's not even being expressed. For whatever reason, it's a defective, and we know that the default color here is white. And so we know that if we have a homozygous dominant, we have two purples, we have big P, that represents this allele with that particular DNA sequence. We have heterozygous, right? This is also going to be purple. And then we have homozygous recessive, which are going to be white. It's pretty straightforward. So when we say the genes are present as pairs, that's really alleles, right? And we get one from each parent. So this particular pea plant got this chromosome with the purple allele from, let's say mom, and it got this chromosome from dad with the white allele. And of course, it inherited one from each parent, and this is diploid, right? It's 2N, which is most organisms. And we'll talk about variations of this later. We know that genes come in alternative forms, and we call those alleles, right? So if we're not talking about pea flowers or whatever, then we can say hair color, and those alleles would be maybe black or brown or blonde or red or what have you, right? So we can have lots of different alleles, but they would all code for, say, hair color or flower color or whatever that is. So usually the phenotypic trait is the gene, and then the alternative forms of those genes are called alleles, which again should be a review. We know one allele can mask the effect of another, and so in that case that's called dominance. And in this case, it's not really masking, it's just kind of overpowering it. This isn't present, so we discussed this. Let me move this. There we go. And so those alleles segregate. If this particular pea plant were to create gametes, these gametes, both its purple gamete allele, right, would be one gamete, and it would segregate from the one that was white, which we represent with little p. And we'll just say that this is a male plant, and so it would produce sperm. And then those gametes, right, would fuse randomly to create a new allele pair. So, obviously it would have to have another parent, or if it would self-fertilize, maybe if it selfed, and then these would recombine into what we call a zygote, which is nothing new. Zygote. There we go. So there is two ways to do genetic analysis. One of those ways is called forward genetic analysis, and the other one is called reverse genetic analysis. So old school geneticists used forward genetic analysis. This was really the only way to sort of figure stuff out. It's kind of easier to do reverse genetic analysis now with the way that we can manipulate DNA, as opposed to this forward genetic analysis, which is kind of a fishing expedition. But both are used. This one is certainly cheaper and takes less time to do because you simply introduce mutations and then go back trying to figure out what gene is involved with that, and then you can go ahead and sequence that gene. This could take quite some time to do, but it's still a tried and true method, and so we're going to discuss it because this is genetics. So we're going to try to discover genes that cause some sort of phenotypic variation, and that could be anything, right? So we have wild type, and we're talking about wild type things. We're talking about stuff that's generally found in the wild, right? With the most common thing in the wild. And this changes over time, and we'll talk about that when we start talking about evolution and things like that. But for the most part, let's just say the majority of fruit flies in the wild are going to have red eyes, right? It's super rare to have any other colored eyes. So we would say that red eyes in fruit flies is the wild type. And in pea plants, the wild type would be the dominant type. So that would be purple flowers, for example. And so that's what we're talking about when we talk about the wild type or the common types. And so we're going to identify something that we're interested in. Maybe we're interested in flower color, or maybe we're interested in seed color. Maybe we're interested in the eye color of Drosophila, or maybe we're interested in the shape of the wings of Drosophila, what genes may control the wings, if they're curly, what genes control body color, what genes control the size of a chicken egg, or how big a chicken is, or whatever that might be. And so we're going to look for mutants that affect that particular trait. So in this case, we would look for something that's not red eyes, right? Some other color eye. It could be dark eyes, like ebony eyes. It could be white eyes, which is a pretty common mutation in Drosophila. It could be white flowers. It could be pink flowers, whatever, just something different from the wild type. And now we're going to have to try to figure out, is that caused by a single gene? Because when you start adding in lots of genes, it gets super complicated super fast, right? And we know that when we have, dominance and recessiveness, and we have a single gene involved in that, and that gene is on even a couple genes, and those genes are on different chromosomes, then it's pretty easy. We know the ratios, right? We know that for example, the purple flowers in Mendel. We know that it's going to give us a 3-to-1 ratio whenever we do a monohybrid cross, right? So we're going to make sure that that's due to a single gene, because if it's polygenic, we know that there's going to be a big range. We talked about that, right? We talked about human skin, right? So we're going to have people that are going to have very dark skin, people that have very light skin, and then there's a whole host of different colors in between, and we call that quantitative traits. So there's a lot of variation, right? Even when we're looking at a couple genes, right? We know that we're going to get like a 9-to-3 to 3-to-1 ratio, or something different than our standard 3-to-1 ratio. So that's what we're looking for to determine if it's a single gene. It could even be epistasis, right? So we have two genes working together, and we know from previous lectures that would cause, these numbers to come together, so we get a 9-to-7 ratio, or maybe we would get these numbers to combine and we get a 9-to-3 to to 1 ratio or a 9 to 4 ratio or something weird that wouldn't follow, our standard Mendel dihybrid, which is 9 to 3 to 3 to 1. So we're going to do crosses to find out if it's a single gene. And then once we figure out that it's in a single gene, then we go on a fishing expedition to figure out what the DNA sequence of that gene is. That will tell us where the mutation lies and what amino acid change caused that particular mutation, right? So why did the purple pigment not fold into the correct shape and it's no longer purple? Why did the red pigment not fold into the correct shape so it's no longer red? In the end, we want to understand how these genes contribute to this phenotype, right? The in this case, eye color or flower color, but it could be anything, right? So it could be, here's just some examples, but you could probably think of a million things that you might be interested in. Maybe you're interested in the size of chicken eggs or the wings. This is a lot of Drosophila genetics here. So what color is the pigmentation of the Drosophila? What is their mating behavior? There are genes that cause Drosophila to males to have mating behaviors with other males as opposed to females. Maybe you're interested in their metabolic pathways. How do they break down sugars? Fructose, glucose, galactose, whatever. Maybe you're interested in, do these genes increase your intelligence or your ability to learn? Or, we're focused on eye structure right now, but it could be any kind of gene, anything that you're interested in. So you're going to try to induce mutations that cause something different from the wild type into your organism of interest. And so when you do this, you want to choose an organism, and usually geneticists choose organisms based on two things. One, what is their development time? Right, so Drosophila are great because their development time is like 5 days. So they could go from an egg all the way to a breeding adult in 5 days. They also have many offspring. And so these two things, development time and numbers of offspring, are things that make organisms ideal genetic candidates. Humans are terrible because their development time is extremely long, years, right? And they usually have very few offspring. So you might not see certain traits in families at all, for generations. And so that doesn't mean we can't study humans, but there's certain things that make them difficult to study, and this is it. So to study humans, we have to construct pedigrees over many generations to try to figure out what if this is something that runs in families, and then what is the mode of inheritance? Because we can't just look at 100 offspring and then see, okay, yeah, there's a mutant here in this human family. So we can sit around and wait for spontaneous mutations. We talked about those, tautomeric shifts, mistakes that the polymerase makes by adding the wrong nucleotides. It's not a perfect system. You're going to get mutations in your DNA, and there's nothing you can do about it. It wouldn't matter if you lived in a lead-lined box. And then there's things that we can do to increase this rate of mutation, right? So we already talked about this in the last lecture. We know that we can X-ray stuff that causes chromosomal breakage, mutations, depurinations, depyrimidations, all kinds of stuff. We can add chemical mutagens, 5-bromouracil as an example. We talked about DNA intercalating agents like ethidium bromide and acridine and things like that. And we know that ultraviolet light causes thymine dimers that can induce mutations as well. and especially when we invoke the SOS system in bacteria, you get tons of mutations from that. So let's say we're interested in the eye, right? And this is just an example, right? And so we do this. We add mutagens. We feed it 5-bromouracil or expose it to radiation or whatever to try to induce mutations. And we're going to get lots of mutations. We're going to get lots of death. We're going to get things that are going to be non-survivable, things that we're not interested in. Maybe it changes the body color. Maybe they get an extra antenna or extra feet. Like that's not what we're interested in. We want to know what controls eyes in Drosophila, the shape of the eye. And so we're going to keep mutating Drosophila till we get something that affects the eyes. And in this case, we discover a mutant that has small eyes, right? And then we're going to have to name it. In this case, it's called eyeless. We'll talk about the nomenclature in Drosophila because it's much different than it is in, say, Mendela, where we have the capital letter. Like in purple, it's capital P because that's dominant, because purple's dominant, and little p is white. In Drosophila, we use a plus system to indicate the wild type. And I'll show you that in a second. So we're going to do the standard cross, right? We want to know, is this in a single gene, right? We have a mutant. We're going to assume that this is going to be a mutation that's going to occur in both of the chromosomes, right? So it's going to affect, let's say that this is eye shape, right? And both of these are broken for whatever reason. And so we would symbolize that by with the lowercase and also with the no plus designation, where wild type would be both plus for a functioning product, right? That makes the eyes normal. All right. So what do we want to know? Is it in a single gene, right? We have to do crosses for that. We're looking for the 3-to-1 ratio. We don't want anything weird like 9-to-7 or 1-to-2-to-1 or this wide range of any phenotypes, because then that would tell us that it's not a single gene. And then we got to find out, is this a dominant mutation or is it a recessive mutation? And so we know that if it's a recessive, just like in Mendel's world, right, we'd cross these and, we could do the Punnett square. Big P, little p, big P, little p. We're going to get homozygous dominant, heterozygote, homozygous recessive. In Drosophila, we would have something like this. And then we would have in the first generation, and so this would be the parental generation. Then we'd have to do the F1 generation because these would all be heterozygotes. And then we would do the next generation, which would be we do the Punnett square. This works the same way. It's just the nomenclature in Drosophila is different. I don't know. I'm not a Drosophila geneticist, and I'm not a big fan of this nomenclature. It seems like a little excessive, but this is how they do it. So this is what we do. So this would be the one with the eyeless feature, right? This particular mutant. These would be heterozygous, so they would look normal, and then this would be homozygous dominant, and it would also look normal. So we would end up with a 3-to-1 ratio. Regular eyes to eyeless. So we do it, right? And I just did the cross, and we get all wild type F1 generation like we showed. And I showed you this nomenclature and did the Punnett squares already, so you could see that. So we have to cross the F1s. And we do that, and then we're going to get, the single homozygous dominant, which I just showed you in the previous slide, we'd get 2 of the heterozygotes, which would, so we add the 1/4 with the 2/4, that gives us 3/4, and then 1/4 of these would be our mutants. So that would tell us not only that it's in a single gene, because we get that 3-to-1 ratio, but also that it's recessive because we get that 3 with the wild type, and the 1/4 with the, so 3/4 of the wild type, 1/4 with our mutant condition. All right, so like I said, Drosophila genetics is different. The nomenclature is different. And I don't know why. It just is. So we just have to go with it. So geneticists, especially Drosophila geneticists, seem to have a sense of humor. And so they name things that they think are funny. They call this one eyeless, right, like we said. And so that's generally represented by a short symbol, usually the first couple letters of whatever they've decided to name it. The symbol is lowercase, right, no matter what. So generally they use the lowercase here if there's only one mutant allele, and then in this case they would add a plus to show it's wild type, like just what we showed. And then if there's more than one allele, right, then they would use it as the superscript, say 1, 2, 3, and so on. All right, so this is review. We know diploid organisms are 2N. That means they have 2 homologous chromosomes, one from mom, one from dad. These are artificially paired up. This is called a karyotype. Karyote is nucleus, and of course they're typing this. So this is a chromosomal squash, and then this is called fluorescent in situ hybridization, which I think we already talked about. And we talked about the chromosomes are numbered by their length. So we have 1, 2, all the way down to 22. We talked about this as a misnomer. This is actually the shortest one, not 22. We figured that out after we were able to sequence the DNA. But they didn't change it. I don't know why, but that just is how it is. And so we have 22 pairs of what we call autosomes, right? These are found in both males and females. It doesn't matter. And in a normal individual, this would be a diploid. We would have 1 of each of our number 1 chromosome, right? And let's say that this has, I don't know, let's say that this is flower. So this might have a purple allele, and this might have a white allele, and then the locus would be the location that it's found on. And then, when this underwent M phase through the cell cycle, these would be each of these would be held together by a centromere. And then if it was meiosis, we would have crossing over, and these would form a tetrad. So anyway, and then we have sex chromosomes, and sex chromosomes generally determine the sex of the individual. We can have XX, right? Usually that's female. It is certainly female in humans, unless there's a mistake in a gene called SRY, and we'll talk about that in a second. And then we have XY. Y chromosomes are much smaller, and that's found in males. So we have 22 autosomes and 1 pair of sex chromosomes. Females have 2 X's, so this is a female. This is a normal individual. Like if this had an extra 21, then we could say that would be a Down syndrome female, right? Because that's called trisomy 21. And then this is a karyotype of a male. Again, this is a normal male. Here's the X and the Y. So that tells us that this is going to be a male. And then all of these have individual pairs. And so this is completely normal. If this had an extra 21, then this would be a male Down syndrome offspring. There are instances where you can have extra of these other chromosomes, but usually they're not survivable, so they end up being a miscarriage. And just a side note is that you can basically determine the sex of your child if you did an in vitro fertilization. What they would do is they would take the sperm, right, and they would centrifuge them down, and so you would get layers, right, and of both sperms that carry the X chromosome and ones that carry the Y chromosome. So, and I actually got that backwards, so let me erase this. So we would expect the Y to be at the top and the X at the bottom. Now, why is that? And the reason is because the Y chromosome, gametes with the Y chromosome are going to be lighter, right? Because the Y chromosome is smaller, it's not as heavy. So in a centrifuge, it's not going to go as far down as an X chromosome. So if you want a male baby, you take the sperm from the top of the centrifuge test tube. And if you want a female, you take the sperm from the bottom of the test tube. And generally that works out. Then you fertilize it, right? So you have your Y sperm, and the mom is always going to have an X egg. And so when these fuse, you end up with a male. If you wanted a female, then you would take those, and you would end up with an XX. So that's how you can kind of pick the sex of your child if you're doing IVF. All right, so here we go, talking about the difference between X and Y. So X is pretty huge compared to the Y. There's 1,000 genes, give or take a few, on the X chromosome, about 150 on the Y chromosome. It's about 3 times as large as far as base pairs. The roles of the X chromosome, because it has so many genes, there's lots of different phenotypes. They generally have nothing to do with sex, and we'll talk about that in a second. Y chromosome is usually male determining. It also affects fertility, and we'll talk about that now. So there are instances where you have chromosomal abnormalities in the Y chromosome in the particular gene called SRY. So I don't know. Back in the day, you had like these East German women who had a lot of male characteristics, winning lots of Olympic events. And so they used to do chromosomal testing, right? So that they would they do a fish, right? Just like this, a chromosomal squash, and do a karyotype. And they would look to see if that athlete was indeed a male or a female based on its chromosomal makeup. Does it have 2 X's or XY? That individual. And in some cases, you could have someone that's XY, but not necessarily a female. And this is why. So there's a gene on the Y chromosome. That gene is called SRY, right? It is in a normal individual, that SRY gene is found on the Y chromosome and not the X chromosome. However, this gene could get translocated to an X chromosome. And so you could have a female that's an XX with an SRY gene on it. And this female, because of that gene would be active, would develop as a male, even though it has chromosomes that indicate a female makeup. You could have an individual that has a male pattern of chromosomal inheritance where it's XY, and the SRY gene is missing or broken or mutated, and no matter that it's XY, it would still develop as female because this gene determines sex, right? It's not the chromosome, it's only this particular gene. I don't want to update you, no. Okay. So, SRY encodes a gene called TDF, testes determining factor. And like we said, if it's present, it produces this protein that causes testes to be formed. If it's absent, that protein isn't made and ovaries are formed. And so this is kind of an interesting scenario because just like we talked about in plants, right, where the flower color is white by default. And so unless there's a gene that may encodes a purple pigment, if that is broken, then that flower is going to be white, right? The same case is true here. There is a default pathway for humans, and it has to do with sex. So if that SRY gene is, like the purple gene, broken, then the default pathway is female. So we are all predetermined to be female, and it's only due to the presence and activity of the SRY gene that causes maleness. So when we have chromosomes aligned, when we talked about this, when we have our homologous tetrads, right? So these are sister chromatids. Let's just call this number 1, and let's say that this is from dad and this is from mom, and they've duplicated in S phase and they're held together with a centromere. So these are sister chromatids. And by that, I mean this is a sister chromatid, this is a sister chromatid. These together, so all 4 of them, are homologous. Because we could have different genes, right? We could have brown hair here, and this would obviously be brown hair. And then we could have blonde hair here, and then obviously this would be blonde too, because those are sister chromatids are exact copies that are made during S phase. These things line up as tetrads, and remember we have crossing over, so I'll change the color. We have crossing over. I'll just make dad's chromosome green. And then mom's chromosome would be red, so we would have her cross over here. And it's because these are homologous, right? The DNA sequences are very similar. That's not the case for the X and Ys. So they're different links, they have different genes on them, but they still line up together, just like all the autosome chromosomes. And the way that they do that is because they have homologous regions on their ends, on their telomeres, that allow them to do this synapsis so that they move together during meiosis. All right. So outside of those regions, they're not homologous. Inside those regions, they're homologous. All right. So that's in humans. In fruit flies, they also have an XY. I'm not going to get into any depth in this. These don't actually have like an SRY gene on it, so it doesn't really code for maleness. But we're just going to, for the sake of simplicity for this class, and if you want to learn more about this, you can take a molecular genetics class. But we're just going to say, for keeping it simple, stupid. I'm not calling you stupid. I'm just saying that, that's the expression. So, just like mammals, Drosophila have an X chromosome and a Y chromosome, but it doesn't determine sex, and I'm not going to really get into that. let's just say for the sake of argument it does, because it kind of, it has to do with the number of chromosomes that determine the sex, not the presence of an X or Y. But that doesn't matter because this is an odd number, this is an even number, and so it's still going to follow the male and the female ratios if it has an XY or an XX in Drosophila. And that's all I'm going to say about it. Okay, so genes that are in different regions, right? They're on different of the sex chromosomes. So you might have a gene that's on the X chromosome solely and not on the Y. So let's say there's a particular gene here that's not found on the Y, or maybe there's a gene on the Y that's not found on the X, and we're going to say that that particular trait is sex linked, right? It could be X linked because it's on the X chromosome, or Y linked because it's on the Y chromosome. Because males only have a single copy of X, right? They don't have two copies of all of the genes. We call that hemizygous, right? They only have one allele. They don't have two. Females would have two of that particular allele, right? And so those would be homozygous. Because of that, so if you have a defect in, let's say, in this particular gene, one of these codes for factor 8, which causes hemophilia. So if this is broken in a male, you don't make the factor that clots your blood and you become a hemophiliac. There's no other X to rescue you. In a female, if you had one of these broken ones, you're okay because this can save you, right? You only need half of it. Your blood is going to clot slower. You're only going to make half the amount of factor 8, but it doesn't matter because you're not going to bleed to death. Whereas in a male, it has no rescuer. You don't have two X's. And so males show the recessive phenotype even if they only have one copy of that recessive allele, due to the fact that they're hemizygous for that particular trait. All right. So X-linked traits were first demonstrated in Drosophila in fruit flies, and it had to do with the mutation in eye color. So white eyes versus red eyes. And this is kind of how it was discovered. So we have white-eyed males, right? And so remember, it's going to follow the same sort of thing. So we're going to have X and Y for males. These are females, so we're going to say that this is XX. And so in this case, it's X-linked, and so this is broken, right? And by default, it's going to be white. And this is going to be wild type, so it's going to be normal. And so it's going to be red, and this will also be red. And I think that I have a slide. I think the next slide shows this, but I'm just going to kind of draw it out here anyway so that you can kind of follow along. So we do the mating, right? And you can do this just like you would do a normal Punnett square. We'll just put this as W for white. Here's our Y chromosome. And then we'll do mom. So we'll do this as dad. That's dad. This will be mom here. So we'll call this R for red. Red. And then we're going to have WR. And this is going to be a female. So this female, to put it on here, this female is going to have red eyes, right? Because even though it has one white, this red is going to overpower that. So it's going to be a dominance thing. This one is going to be red from mom, and then this will be white from dad. So this is also going to be a female with red eyes, right? So we're going to get 2 fourths are going to be females with red eyes. And then here we're going to have, this is the red eye. This one is going to be Y. So this is going to be a male with red eyes. And this is from this one here. And then this is the Y. So this is also going to be a male with red eyes. And so we're going to be 2 fourths of these. And, we reduce this to 1/2. So 1/2 are going to be females with red eyes, and 1/2 of these are going to be males with red eyes. And that would look, this is going to look normal to us, right? Like we would expect this, even if it wasn't sex linked. They should all, no matter if they're male or female, they should all have red eyes because red would be dominant to white in Mendel's world. But it's not until we do the F2 generation cross that we actually see a really weird ratio where all the females have red eyes, only half of the males have red, and the other half have white. So this is going to show you what we just talked about. So this is going to be the female, right? This is going to be representing the X chromosomes. So W plus is white eyes. Remember, W is the trait, the mutant trait, and then we use the plus symbol to show that's wild type. So W plus would represent the red allele, right? That's going to make a protein that's going to transmit the red wavelength to our eyes. So when we look at the fruit fly, its eyes will be red. And then we're going to have the male, right? It's only going to have one copy of this, right? Because it's X-linked trait. And so we have this male that's going to have a broken one. And when it makes the protein, it's not going to fold into the correct shape. So it's not going to transmit red to us. That by default, it's going to transmit white to our eyes, right? So white-eyed male, red-eyed female. And then, so this one, this is going to be a Punnett square. So this is the F1 generation. This represents the male, right? Here, so we have the male can give this gamete or this gamete, right? And then this is going to represent the female here, and it doesn't matter which one it is. The female can only give the red X. And so when this white X sperm meets this white X egg, we're going to get red-eyed females. This is going to be the Y chromosome, right, from this male. This is the only egg that this female, so let's just put tails on it to represent the sperms. And so we're going to get red-eyed males, right? So all the offspring are going to be red-eyed, males and females. Now, these are going to produce, when the F2 generation, so this is the female, this is the male. So the female can produce, right, has a half of its gametes, right? Half and half, which would be a quarter, right? So we're going to be the red X chromosome, and then half will be the white chromosome. This one is the male gametes, so we'll put tails on them just to represent this is it. So this one is this one. This one is this one here. And then we do the cross. It's like a standard Punnett square. So in this case, this is going to be X at both red. So this is going to be a red-eyed female. This one is going to be the X chromosome with red on it here. and the Y chromosome with nothing. And so this is going to be a red-eyed male, right? Because it's XY. This one is going to be a red from dad, right here, and then white from mom. And so this is going to be a heterozygous, right? Mutant, wild type, red-eyed female. And then here, we're going to have the white male. So white-eyed male. So only males in this cross are going to have white eyes. All of the females are going to have red eyes. Half of the males will have red eyes, and half of the males will have white eyes. This is half of the gametes, right? So I don't like this the way that this is drawn out in the text, but what this quarter is representing is 1/2 times 1/2 is a 1/4. So 1/4, this 1/4 is going to be a red-eyed female, and then this 1/4 is going to be also a red-eyed female. So we add them together, we get 2/4 or 1/2. So 1/2 of all the offspring, 100% of the females, half are going to be 50% of all the offspring are going to be female, and 100% of them are going to have red eyes. And then again, this is 1/2 times 1/2 here, which is 1/4 overall, are going to be red-eyed males, and then 1/4 of all the offspring will be white-eyed males. Now, 50% of the males are going to have red eyes, and 50% of the males are going to have white eyes. And we can go through this same cross here. I'm not sure I want to go through the whole thing, but there are some practice problems for you guys to do this. But you just do the standard nomenclature. Just make sure that you represent the Y chromosome as having no allele on it, and only the X chromosome has the allele. So in this case, this is a red-eyed male. Remember, it's hemizygous. It only has one allele, none on the Y chromosome. That this is an X, this is a Y. This is a female, so it has two X's. Both are W's, right? This one can only produce the gametes. Female can only produce white. And the male can produce only red, right here. And then this is only the Y. And then we can follow the crosses out, right? So half of the males are going to be white-eyed, and half of the, or 100% of the females will be red-eyed. 100% of the males will be white-eyed. And we would expect a 1-to-1 ratio, right? Half of all the offspring would be female, half of the offspring would be male. 100% would be of the females would be red-eyed, and 100% of the males would be white-eyed. And then we can carry on this cross here. So it really depends on how we do the cross, right? If we start with a red-eyed male or a white-eyed male. And then we get these weird numbers, and it's due to standard Mendelian genetics. It's just that there's only one copy of that allele in males. Okay, so hopefully that makes sense. And like I said, there's a lot of practice problems that you can do in the problem sets. And I'll put some of these practice problems in the quiz for this particular chapter as well. So X-linked inheritance. There's key features to that, right? Males inherit X-linked traits only from their mom, right? Because when you have a male offspring, the mom always contributes the X chromosome. The dad always contributes the Y chromosome, right? Dad can't give you an X because if he did, mom can only give X's. So if your dad contributed an X, you would be a female. So all of the X traits come from mom. Males don't pass on X traits to their sons, right? That can only go to daughters. All daughters of an affected male carry the allele for that trait, right? Because if you have a mutation, let's say you're a male and you have a daughter, well, that mutation has to get passed on to your daughter from you, right? Not from the mother. So here's some examples of some X-linked disorders in humans. This is a 6, by the way, in case you're colorblind, you can't see that. So these are recessive ones: hemophilia, which we talked about, factor 8, Duchenne's muscular dystrophy, testicular feminization. Remember we talked about well, we didn't really talk about this, but this is a condition that they think Joan of Arc may have had. We have color blindness, which is why I put that in there. And then there are dominant X-linked disorders too, which are super rare, like vitamin D resistant rickets. There are some Y-linked mutations as well. It's very simple pattern of inheritance, right? I mean, if you're a male, right, and then you're an XY, right? So if you pass on an X, moms can only pass on an X. So if you pass on an X, that's a daughter. If you pass on a Y, that's a son, right? So the phenotype is passed only from father to son, right? Whatever is on this chromosome is going to be given to their son. There's no other way that the inheritance occurs here. All the male sons inherit this phenotype, right? So no matter what mutant is on here, it's also going to be inherited by their sons. None of the daughters do. The reason is because you cannot give any Y-linked chromosome to a daughter. You can only pass on your X. Otherwise, it wouldn't be a daughter, it would be a son. So SRY gene, right, is on the Y chromosome. We talked about that. There's some fertility factors that can cause you to be infertile, and they're related to mutations in the Y chromosome. There are some other non-sexual traits, but there aren't many examples, or they're too complex to actually say that they're single genes. Okay. All right, so there's things called sex influence traits that aren't necessarily sex linked. Male pattern baldness is one of those things. So it is expressed differently in males and females. There is some genetic variance, but it's probably more autosomal than it is linked to the X or the Y, the sex chromosomes. And then, in male pattern baldness, this is related to high levels of testosterone that lead to that, rather than like something that's found on the Y chromosome or the X chromosome. Okay, so that leads us to other variations in inheritance. And in this case, we're talking about organelles. So hopefully you learned this in intro bio, but you know that mitochondria have their own DNA and chloroplasts also have their own DNA. And because they have their own DNA, they have their own set of heritable traits that are passed on from parent to offspring. So mitochondria and chloroplast are generally circular chromosomes. We talked about circular chromosomes versus linear chromosomes, and you should know from at least this class that usually it's prokaryotes that have circular chromosomes. And so this is a good indication that these organelles are more closely related to prokaryotes than they are eukaryotes, would typically have linear chromosomes. The other thing is mitochondrial chromosomes are generally much smaller than chloroplasts, which are usually a lot larger. even though they're some like the mitochondria range from 6,000 to 2.5 million base pairs. That's for the whole chromosome. Humans have about 16,500 bases of mitochondrial DNA. Not a lot of genes, especially considering we're 3 over 3 billion nucleotides in our nuclear genes, in our nuclear DNA. Chloroplasts are bigger by comparison to mitochondria, but they're still relatively small when you compare it to nuclear genomes. And if you just want to look at humans, the smallest chromosome, remember we said it's 21, not 22. That's about 47 million base pairs. So we're talking orders of magnitude larger than mitochondria. This is just showing you that euglena, this is a protist, and it's been stained. So its nuclear DNA here is red, and its mitochondrial DNA are green. And you can see that there's lots and lots of mitochondria per cell, right? So there's only one, and because it's a protist, it's a eukaryote, so it has a nucleus, and that means it has nuclear DNA. So there's only one copy of its nuclear DNA, but it has lots and lots of mitochondria, and so that has lots and lots of copies of its mitochondrial DNA, which we just talked about. So organelles have lots of copies, right? You can have, hundreds of mitochondria in a muscle cell, whereas in that same cell, you would only have one copy of your nuclear DNA. Each organelle has 20 to 40 copies of its genome. It's still relatively small, right? Less than 1% of nuclear DNA, even if we add up all the organelles all together in a single cell. And the genomes encode genes that are related to that particular organelle's function. So if we look at humans and we look at their mitochondrial genome, there's 22 protein coding genes in all of that genome. All of those genes in a mitochondrial genome function to do things in the mitochondria, right? Chloroplasts, right, have 100 genes, but those proteins participate in the process of photosynthesis, whether it's the light reaction or the dark Calvin cycle. Those genes are dedicated to photosynthesis. These genes are dedicated to the production of ATP in the mitochondria. And again, this is, kind of what we think the origin of organelles, especially ones that contain DNA like mitochondria and chloroplasts. Probably some sort of symbiotic relationship where, this cell gave it protection, this produced ATP for the cell, and they lived happily ever after. And, at some point, this is a plant cell because it has mitochondria and chloroplast in it. So, now it can produce its own food. It doesn't have to go around the environment and get its own food. So here's the genes. Some of these code for tRNAs. Remember we talked about the different ribosomal RNAs. This is similar to the prokaryote, right? Ribosomes as opposed to the eukaryotes subunits. And so that's another telltale sign that this is probably related more to prokaryotes than eukaryotes. And it has that circular chromosome that's generally only found in prokaryotes. And a lot of these genes, like cytochrome B, that's involved in the electron transport chain. Okay, so hopefully everything there is to know about organelles, at least the ones that contain DNA. And now we're going to talk about how this inheritance occurs. So the organelle inheritance. So, well, I'll just go on with this. So there is a flower. They're called 4 o'clocks, and they can have white leaves, and they can have what we call mottled leaves, and then they can have pure green leaves. And so scientists were like, well, what's going on here? And they did some crossbreeding, and they crossbred a male to a female, and all the offspring, whether they're male or female, were green, right? So this, you would say, okay, well, this is just a straight up dominance inheritance, and what's the big deal? But when they do the reverse, where they have the male that's green and the female that's white, all the offspring are white. So what's going on here? And so the white color is due to the chloroplast, right? We know chloroplasts do the photosynthesis, and that contains chlorophyll, which has the magnesium heme that causes the electrons to go through the electron transport chain in the photosystem 1. Photosystem 2 is before photosystem 1. And so chlorophyll is a green pigment, right? It doesn't use green wavelength. That's reflected to our eyes. It uses almost everything but green. And then these are chloroplasts that are white. And so if we look at the cell, remember we talked there's lots of organelles within the cell. This is the nucleus. So most of these chloroplasts here have a white color. Most of the chloroplasts here have a green. So if they're green, like here, then the leaf is green. If they're white, like here, then the leaf is white. And when they're like this, some of the individual cells in here have green chloroplasts and some have white ones. Now, why is that? All right. So in general, and then this isn't always true, but most of the time it's true. Offspring inherit organelles generally only from their mother. And the reason is because if you look at a sperm or pollen or whatever, the sperm is generally very small compared to the egg, right? Because the sperm is so small, most of all of the material that's in the sperm is DNA and ATP, and that drives the tail. Same's true with the pollen. Usually there's lots of pollen. They're very small. They don't contribute many organelles at all, right? In fact, if you looked at humans, there's no mitochondria is contributed to the sperm. So all of the organelles, right, especially the mitochondria and plants and chloroplast or whatever, they all come from the mother, right? So when this is fertilized, no organelles come from the father at all, only the DNA. And so whatever the mother's mitochondria are what you inherit, right? So in this case, the mother produces the egg with all of the chloroplasts, right? And so her offspring are going to all be green. Where the father is white, he contributes hardly any, if at all. And so even if there were a couple in here, it's still going to look green. If we switch this, right, where the mother, again, she's going to produce the egg with all of the chloroplast, right, because the father's pollen is very small and there's not a lot of room for any extra material besides the DNA, right, the nuclear DNA. And so the zygote, the fertilized egg, is only going to have mitochondria from the mother. The father doesn't contribute any mitochondria or chloroplast in this case, because we're talking about plants. And so all the offspring are going to be white, like the mother. So we call this homoplasmy, which means all the cell's mitochondria have the same genotype, right? All of these are white, all of these are green. But we can also have a condition called heteroplasmy, where it's a mixture of distinct mitochondria or chloroplasts in this case. So we have some chloroplasts in this cell are white and some are green. Now, if there's more white ones than green, it's going to look more white. If there's more green ones than white, then it's going to look more green. And you can have, remember, these cells are going to make new cells. And when they do, you can't make chloroplasts from nothing. So these are going to get divided in a certain way, right? And so it could happen where once you go through this cell division, and maybe this is how the chloroplasts line up for this particular cell division, and so only white ones get passed on, and then the other cell gets a mixture. And you could have the same thing going on here, where you just have like the daughter cell here would, I mean, it's shown as a mix, but it could just be all green too. And so this is just kind of another graphical representation. So if we're just talking about a couple mitochondria, we have daughter cells. This one gets divided. Maybe this becomes homoplasmy, heteroplasmy. And in the end, all of the descendants are going to be white. And here we'll have, because green is going to overpower white, so these would look green. These would be pure green. All their offspring would be green. And the same thing with mitochondria. It's the artist is just showing you that these mitochondria have a certain genetic composition. This one is different, and so you could have some that are end up with all of the same mitochondria, all genetically identical here and here, or a mixture, right? After several rounds of cell division. All right, I think I already jumped ahead and talked about this, but we want a short generation time, right? So fruit flies are good. For plants, Arabidopsis is good. C. elegans, it's a nematode, is good. Mice are good. Humans are terrible. The reason are they have lots of offspring, right? And the other thing I didn't mention is it's really easy to arrange a mating. So if you want a big mouse to breed with another big mouse, you just pick the two mice and put them together, and then they mate and have offspring. Same thing with the fruit flies. This is a little more difficult because they have such a fast gestation period. You have to like knock them out with the carbon dioxide, and then you can select virgin fruit flies from others, and that's kind of a pain. But mice are a good model. Like I said, I'm not a fruit fly biologist, but there are plenty of people that do that. And if it wasn't for them, we wouldn't know half of the genetics that we know now. So we talked a little bit about that. In humans, we don't have any of these advantages. We don't have a short generation time, right? Like we're talking, 15 years, maybe 18 years. Usually humans don't have a lot of offspring. We're not talking about hundreds or thousands. And you can't force, it's unethical to force people to mate with each other. And, if you're trying to figure out, like, how is this trait inherited? So in order to understand human genetics, we have to take a different approach, especially for, heritable diseases. And so the way that we do this is we use pedigrees. So these are just examples. We're going to talk about polydactyly later. Sickle cell anemia we talked about already. Huntington's disease, breast cancer. This is kind of interesting because there's genes called BRCA1 and 2. And these, they don't guarantee that you're going to get breast cancer, but they make it one step closer. So we talked about cancer before, and we talked that there's at least 4 things that have to go wrong. We had to have tumorigenesis, we have to have metastasis, we have to have angiogenesis, we have to have immortality. And let's say that there's 10 steps to get cancer, right? And you could inherit 3 or 4 broken genes, so maybe you're 6 steps away, right? And so this is one of those genes that will make you one step closer to getting cancer. And maybe you smoke, and so that causes 2 or 3 more mutations, so maybe you're only 3 steps away. And maybe you live on a uranium farm, I don't know, so you're 1 step away, right? So just because you have this gene doesn't mean you're going to have breast cancer, but it means that you have a higher probability than most people to get to 0 steps, which equals all the bad things, metastatic breast cancer, Tay-Sachs disease, PKU, we'll talk about that, cystic fibrosis, dwarf. There's lots of other ones. This is a dominant trait. So we have to use pedigrees, right? And going back to the breast cancer, this was actually discovered by a nurse who did a pedigree on her family. So pedigree analysis is really important, right? There are some problems with this, but it's really the best way that we have of understanding human diseases. I mean, now we do reverse genetics, so it's a lot easier. We can find what may be linked by looking at the variation between whole genomes of DNA and comparing one genome to another and saying, okay, these letters are different. This person has breast cancer. This gene is probably involved in breast cancer, instead of like irradiating humans or waiting for mutations to occur spontaneously. And we'll talk about how we do that in subsequent chapters. So you might not have the right kind of matings, right? Like, rare diseases might not be kept in families. The easiest pedigrees to understand are things that have inbreeding, because that selects genes into the same population. And a good example of this is the British royal family. There's another one called the Blue People of Troublesome Creek, Kentucky, and I'll show you that pedigree in a minute. So families are small. It's hard to test for Mendelian ratios. Sometimes you don't know who the baby's daddy is, right? There are studies that show that 10% of all humans on the planet who think they know who their father is are wrong. So 10% of paternity is incorrect. And then generation time is very long, and that's obvious. So rules for a pedigree, right? To draw a pedigree, squares are males, circles are females, diamonds mean the sex is unspecified. So this is an unknown sex. This is square, so it's a male. This is a female. And we number generations in Roman numerals. So this is first generation, second, third, fourth, and we would go on and on. And then usually we number left to right as from offspring, just to keep them separated from one another. So this would be 1, 2, 3, and 4. So if I'm trying to describe someone in this pedigree, I would, like, for instance, this female individual, I would say that one is 2-4. And you would know I'm talking about this individual right here. Matings are horizontal connectors, right? So this is a mating, this is a mating, this is a mating. Offspring are linked to the parents by vertical lines. So these are the offspring of these parents here and here. Individuals with unusual phenotypes are shown in black. So this is usually the condition. Like, let's say that this is hemophilia. Just for fun, or maybe it's a blue person. I don't know. Double horizontal lines. We call this consanguineous. That's a nice way of saying inbreeding. So this is a consanguineous, cousins or closer mating that produce this offspring, right? And these consanguineous breedings tend to keep rare alleles in a population. And later on, when we talk about getting purebred lines, you can backbreed individuals like mice over and over again. And statistically, I'll show you how after about, 10 or 20 of these backbreedings, you're going to end up with a purebred mouse that has that particular mutation as a homozygous recessive mutation, or it could be homozygous dominant if you're looking for a dominant mutation. The first person who comes to the attention of a geneticist with that condition is called the proband. So in this case, remember we designated these as 4-1. So this is 4-1. Maybe this person had a breast cancer and they wanted to know if that's genetic. You would go to a genetic or a genetic counselor. They could construct a pedigree and say, oh, yeah, this is maybe in your family. And then they would try to decipher what is the mode of inheritance. So this is how we figure this out. This pedigree shows a recessive disease. The feature that tells you that it's recessive is that affected individuals have unaffected parents, right? Even though this is an inbreeding. It's generally this one is autosomal recessive, and we can tell because if it were X-linked, then this individual, so this individual here, would be hemizygous carrier for the diseased allele. And we could do the crosses for this, but I'm not going to go through that. I'll let you guys do that just in the interest of time. But you can go ahead and do the cross. So remember that we have the square is male. This is a female, right? This is, so this would be an XY. This would be an XX, right? If it were X-linked, then this individual, right, would pass on its X chromosome that's broken here. We don't know this one, so we're going to cut that one out. But this individual would be a male, so this would get a Y. This would get a normal, right? This, when we don't know, generally we assume that they're not affected, right? So these would be unaffected individuals. And so this offspring could be a carrier, 50% chance, right? Because it would inherit. This is a female that would inherit this affected chromosome from dad, because that's all dad can give, right? And then we assume that this would be a normal individual, so this female would be a carrier, and this would be so this particular male would have to be hemizygous, which because this female would definitely could get an affected X here, but in order for this female to show this trait, she would have to have two affected X chromosomes. It's impossible for this male to have an affected X because it would show the trait, and this would be filled in. Right. So that's why we know that it's autosomal recessive and not X-linked. Okay, let's see. Let me move this. All right. So Rare recessive traits are sparsely distributed. There's not a lot of individuals with the condition that is filled in here. We talked about inbreeding increases the likelihood of the rare phenotype showing up, which is in this case. And if we just go back to this, instead of making it sex linked, we just make it autosomal, you would see that this person would be a carrier. This person could also be a carrier and not show it if it's recessive. And so, because of this inbreeding, we increase this risk of that particular recessive autosomal trait being shown in that individual. Again, it's hard to detect these ratios. Basically, as a genetic counselor, you're just doing the best that you possibly can. So, autosomal recessive traits, albinism, PKU, sickle cell anemia, Tay-Sachs, this is involved in the lysosome not being able to break down fats properly. Cystic fibrosis, galactosemia. All right, so then we have autosomal dominant traits. So, features of autosomal dominant traits are affected individuals always have an affected parent, right? This is passed on generation to generation. Disease appears in every generation. We just talked about that. And affected parents pass the trait equally to both sons and daughters. So, dominant traits, dwarfism, we talked about that. Night blindness, Huntington's disease, that's a dominant trait. Marfan syndrome, widow's peak, and so on. I'm not going to test you on this. I'm just showing you. You will need to be able to figure out these pedigrees. So I will put some of these pedigrees on the test, and I'll also put them on the quiz, and you'll need to determine the mode of inheritance. I also did put some on the problem set as well. So here's X-linked recessive trait. So we can just do this. So this male has it, right? And this is a female. She's going to be a carrier. She has to be, father has to give her this X. This we assume is a normal individual, and it's shown as normal. So because it's sex linked, we can assume that anyway. And then this is going to be passed on to this male can only give a Y. This would be the X that's contributed, and since there's only one copy of the X, then it's X-linked recessive here. So one key feature is males definitely show this more than females. If we looked at the British royal family or any other like X-linked recessive trait, almost only males would be affected by this. No offspring of an affected male have the trait. All daughters are heterozygous, or they're carriers of the trait, right? Because in order to be a daughter, that's a son. This would be a Y, and this would be a normal. But any daughter that you have here has to get the affected X chromosome from dad, right? So she's always going to be a carrier. This is from mom, and this is from dad. Sons of affected male never show the trait and never pass it on to their offspring, right? Because the mother is normal, and so all males are going to be normal from that male. The male doesn't contribute this. It's only from the mother, right? The father wouldn't contribute it because he would have to have the trait as well, and so this would be filled in. So color blindness, red green, hemophilia, Duchenne muscular dystrophy, and androgen insensitivity. All of those are those linked recessive traits in humans. X-linked dominant traits. So affected males pass traits on to the daughters, but not the sons. So here we only have daughters, right? So let's say that this is the trait. Mom's normal. So dad can only pass on the affected X. Mom cannot. She's normal, right? If she had it, then she would also be filled in. So then this would be dad's X defective, mom's normal. And because this is a dominant trait, that would get it. Here, dad cannot pass this on, right? He can only pass on the Y chromosome. The X comes from mom always. So this male would never have the trait. Only females have it. Now, this female can pass this on to a male because now she has an affected X that she can pass on. She can also pass it on to her daughter. So affected females pass trait on to half daughters and half sons. And that's how we can tell it's an X-linked dominant trait. So we talked about vitamin D deficiency that causes rickets, hypertrichinosis, which causes excessive body and facial hair, and you've probably seen examples of that. This mode of inheritance is very rare, but it still occurs occasionally. Mitochondria trait, remember the mitochondria always comes from the mother, so whatever the mother is, that's what the offspring are, right? So, this is a child, right? We're talking about these are the offspring. This is an offspring. Every offspring is going to have it if mom has it. Only the mother can pass it on to their children. So if this is a mother, all of her offspring are going to have it. This is the mother, all of her offspring are going to have it. This is a father, none, right? Dads don't contribute to mitochondria at all. So it doesn't matter what dad is, only what mom is. So affected females always pass on the trait. Males never pass on the trait. Leber's hereditary optic neuropathy is an example. There's red ragged fiber disease. There's some other ones. All right, genetic counseling involves analyzing pedigrees and estimating the risk that you would inherit these disorders, right? So for example, you might want to know, okay, I'm a carrier of Tay-Sachs. What's the odds that my child is going to have Tay-Sachs disease? And maybe it's 3 to 1, right? Or maybe it's 100%, right? Maybe you would decide that you don't want to have children because they would all get Huntington's disease or something like that. There are some instances, I think there's a Lifetime movie on this, but there was a child who ended up with leukemia. She needed a bone marrow transplant. And it's a straightforward two allele, like one gene outcome. But the parents went to a genetic counselor because they couldn't find a bone marrow donor to find out if they had another child, what the probability is that child would match and save the daughter that had leukemia. They went to the counselor, and it was a standard 1/4, right? Probability of matching. So they said, okay, 25% chance we have another kid. It's a bone marrow donor match. Let's have the kid. They did. They got lucky. That kid was a match. And so they took the bone marrow from their daughter and gave it to their other daughter with leukemia and saved her life. I think it's called My Daughter's Keeper or something like that, if you're interested in it. But genetic counseling is a real occupation. It pays quite a lot, 6 figures. And your job is to estimate likelihood of carrying diseases and how that's going to be passed on. And I've given you a couple examples of that. And then I'm going to finish off this lecture just showing you. So this is methanoglobinemia. It is so hemoglobin becomes methanoglobin when it becomes either it's oxidized, right? So it has to get reduced. So there's reductase. And just to remind you, an oxidation reduction reaction, if you gain an electron, you're reduced, right? So like if let's say we have this A and we have B, and so A is going to pass the electron from itself to B. So this one gains the electron, so this would be the reduced. And this one loses the electron, so this would be oxidized. So anyway, it's just a transfer of electrons, right? And individuals would have this, they have a mutation in this reductase, which causes them to not be able to reduce hemoglobin, reduce methanoglobin to hemoglobin. And so they get a condition that causes their skin to turn blue. And so these are the blue people of Troublesome Creek, Kentucky. And the reason I put this in here is because I wanted you to see this is how a pedigree works and this is how they figured this out. But I want you to see what happens when you have these consanguineous matings in here. So we can just count these. There's 1, 2, 3, 4, 5, 6, 7, 8, 9. And so these inbreeding events cause all kinds of genetic problems because it fixes these alleles into the population, right? So, I mean, this isn't that terrible. It just causes your skin to be blue. In fact, you could take methylene blue and it would help. It helps to reduce methanoglobin to hemoglobin. But it could easily be hemophilia that kills you. And I'm quickly just going to wrap this up. So let's just look up pedigree. I'll just pause this. Okay, so this is the pedigree of the British royal family. Let's see if I can zoom in on this a little bit. I don't want you. Okay. I don't want you either. So this is Queen Victoria. It's not let me draw on this. So I'm just going to use the mouse. Queen Victoria, Prince Albert, right here. Queen Victoria right here. She's a carrier of hemophilia. This is an X-linked trait, right? And you can see that in here, remember usually the affected individual is like a half shaded or a dot in it. And so that's showing you that it's a carrier female. So generally females are carrier, males are the only ones that actually get the disease. You can see all these are males. This right here is the Tsar Nicholas II of Russia. And you probably heard about the Romanov. So Anastasia is in here. And anyway, so this here was the issue with the Russian Tsar because he had 5 children and only one of those children was a male, right? And that male had hemophilia. And so you probably know, like back in the day, you wanted to be a male to be the heir to the throne. And so this guy named Rasputin, which you may have heard of, and if you haven't, you can look it up. Kind of a bad guy. Anyway, he convinced Tsar Nicholas II that if he could cure his son of his hemophilia, and he did it with like blood transfusion and leeches and stuff. But only in return he wanted to have power over Russia. So the Russian Revolution, believe it or not, was actually caused by a genetic disease. And then you probably heard about, all these Anastasia, being all these individuals being beheaded during the Russian Revolution, the Romanovs. And so, anyway, this goes all the way down to. I'm surprised they don't have the consanguineous matings on this. Let me see what I find. Okay, so I couldn't really find a decent one on the internet, so I just went to my Bio181 Intro Biology for Majors PowerPoints and to show you this. So this is Albert and Victoria. You can see that they are consanguineous, right? They're inbred. And then we also have that occurring here. And again, here with Queen Elizabeth II. Yes, that's Prince Charles's mother. And why did they do that? And here's another one. And the reason they did that is because they wanted to keep all of the wealth within the families. So it was very common for individuals related to one another to inbreed so that they would be able to continue on with the power and the wealth within that family. All right, so this wraps up this whole lecture, right? And so I'll pick up with a new one tomorrow.